Hi, my name is Charlotte Bascom, and this is my DNA replication PowerPoint. So I went with a PowerPoint as my animation, which I think turned out pretty good, and you can still fully get the idea. So DNA replication by Charlotte Bascom. So what is DNA and where can you find it? DNA is the molecule in our body and other living organisms that contains our genetic code of organisms. DNA is in every cell of your body, and it tells your cells what proteins to make. So every living organism ever has DNA, and it's like a blueprint of your body and the structure that you have. Most DNA is found in the nucleus and every cell in your body. However, a small amount can be found in the mitochondria. So let's talk about eukaryotic and prokaryotic cells. So I know I have prokaryotic written first, but the prokaryotic cells on the bottom and the eukaryotic cells on the top. Now these can be extremely confusing. It even took me a little while, but here's how I broke it down. Prokaryotic, the meaning pro is before and karyo is nucleus. So this comes before the nucleus. It's a small single cell. The DNA is free floating in the cytoplasm. And here's the big thing. The DNA in the prokaryotic cell, so the DNA is in the prokaryotic cell, which is inside of the cytoplasm. And these have no nucleus. That's the big thing. They have no nucleus and they lack organelles. So going down to this picture down here, it's kind of hard to read. I get that. But if you just take a look at it, you can see this entire red thing right here. This is the entire cell itself. And then if you go a little bit further in, this word cytoplasm, this big blue area right here, this is the whole cytoplasm. And going back to what it says, the DNA is in the prokaryotic cell, which is inside of the cytoplasm. So inside of this tiny cell, inside of the cytoplasm, inside of the prokaryotic cell is your DNA. So that's where your prokaryotic cell is. Now on to eukaryotic. So the DNA is in the nucleus. So this cell has a nucleus. It's a large cell. Some are single-celled and some are multicellular, but they have a nucleus and they are enclosed in what is called a nuclear envelope. So once again, the picture is kind of hard to read because of the words, but these brackets right here on the right, if you look at the right of the picture, the bracket says nucleus. So everything within this bracket has to do with the nucleus. So if you look directly here at this nucleus, this little ball in the very center, that's your nucleus. And so that is where your DNA is inside of this nucleus. And then if you look at the circle just a bit above it, obviously it's an opened circle so you can see inside of the cell. But if it were closed, that is your nuclear envelope. So inside of this cell, inside of the nuclear envelope, inside of the nucleus is your DNA. So you can kind of get a picture of how small your DNA really is. With prokaryotic, it's inside of a cell, inside of the prokaryotic cell, inside of the cytoplasm. They're very, very small, but you have tons of them in your body. So now let's break down physically what DNA is. So you have so the base component for each species will always be the same, will always be different. So for example, my base component is going to be different from a monkey, is going to be different from a plant, it's all different. And then there are three parts to the DNA nucleotide. There is the sugar, there is the phosphate, and there is the base, which we're going to go over in a second. But to put it simply, the base, you have A, T, G, and C. So now let's talk about your base pairings, your complementary base pairings, C and G and A and T. These first lines right here, C plus G, A plus T. Getting into actual DNA and how the ladder works, we're going to use the example of the ladder. It's just a little bit easier to understand. You have your bases. These consist of the four letters above, A, C, G, and T. The letter pairings do not change. C will always be with G and A will always be with T. They do not change from each other. C, G, A, T. Remember that. A and G are bigger strands, while C and T are smaller strands. So one big strand goes with one small strand. So if you have the pairing A and T, your A strand is bigger and your T strand is smaller, but they go together. Same with the C and the G. Now I have an example here at the bottom. You can see it looks just like a bunch of letters, and it kind of is just a bunch of letters. But if I bring up the one below it, you can see how they pair up. So starting with this first A, right below it is a T. And then you have two T's next to it. You have two A's on the bottom. They always match up. A's, T's, G's, C's, the other way around. They do not change. You will never see a T with a C or a G with an A. They just don't work that way. So now let's get into the ladder. Like I said, we're going to refer to the model of the ladder instead of the DNA double helix. It's just a little easier to understand. But obviously, when you think of DNA, you think of this double helix strand, which is exactly what it is. But if we're going to break it down, we're going to look at this ladder. So you have the sides, the poles, the ladders, and the rungs. So the sides and the poles are basically the same thing, but what you have is you'll have 3 and you'll have 5. So you can see how we call this anti-parallel. Well, you'll have, instead of it being 3 on the top and 5 on the bottom, you'll have 3, 5, 5, 3. It does not matter which side your 3s and your 5s are on, but it does come into play a little bit later. So your sides and your poles, so the very sides of your ladder right here, are your sugar phosphates. The ladder itself is made up of bases, and the rungs are made up of those bases. So now let's get into the plan. So in order for living things to survive, they have to have 3 things. They need the energy, a plan, and the parts to make themselves. In order for your body to keep making DNA, which is your blueprints, which is how your body keeps making itself, you have to have a plan. You can have the parts and you can have the energy to do it, but without the plan or the blueprints, your body will not know how to replicate itself. If somebody told you to build a building and you had no instruction on how to do it, you could have all the equipment and you could have a ton of workers with energy to help you every day, but without a plan, you don't know how you're going to make the building. So now let's get into the physical form of the DNA strands. So your DNA strand will start out looking like this. Once again, we're going to use the ladder. So this is how your DNA strand will start to look out. Notice how all the A's match up with all the T's. So across from the ladder in the first rung, A and T, and then it goes G and C and all the way down, but they match up with their complementary base pairing. Also notice how they are on opposite sides of the ladder. This is very important. So now your DNA decides that it needs to replicate itself and it needs to make a copy. You have to have an old strand and a new strand. So what does this do? It splits itself in half. This is also known as unzipping itself. All of your DNA strands will do this. They all double. Your 2 strands are known as your old strands. So going back to this last slide, here is your ladder. It's completely intact. It's one whole picture. But in this next slide, they are split in half. I color-coded them into red and blue. It will help a little bit later in future slides, but they are split in half and they are both old strands. The letters from each side of your ladder will stay on the ladder. So in this last picture again, it says the letters are on opposite sides of the ladder, and that is because when it gets split in half, each letter on each side of the ladder stays with that ladder. It has to stay there. And also notice again, we have 3 and 5 and 5 and 3. Now this individual strand is exactly what you need. This strand still has its letters attached to it. This is the template side. The other half will meet up with the bases and be added in later. This is the old strand of the DNA. So you have your letters here, they stay here. This is the old strand. This is like your blueprint. So we're going to get into what is an enzyme and why is it important in DNA. An enzyme is a protein that makes reactions happen. In other words, it's a catalyst. All enzymes are catalysts, and it's important in DNA because it acts like the zipper and separates the DNA strands. This specific enzyme is called a helicase. So you can see the helicase here. It's obviously not in a zipper form, but it is going through and it is separating your DNA. The helicase is like a zipper, and it's what separates your DNA. And then down here, this little orange circle is your enzyme, and it's looking for a substrate. So that's how an enzyme works. But this helicase up here is very important. It's like the zipper, and it unzips your DNA strands. So like in this last slide back here, you have 2 separate sides. This is very important. So what happens to the other half of your ladder? Because obviously it's split in half by your helicase, so you have to have a completed ladder in order to have DNA. So what do? Your new bases will come in and match up with the correct basing on the existing old strand of the DNA. So you can see here we have them split in half. Now keep in mind, remember again, you have 3 and 5 on the left side and 5 and 3 on the right side. Once again, with your letters on the other sides. So one by one, your letters will start to come in and pair. Notice how they pair with their corresponding letter, G's and C's, A's and T's, and they come in and they start to pair up. Then your ladder will close. But there's a problem here. Both of your letters are still on one side of the ladder. What happens if they need to split up again? One side is left without any letters. So what we do is we put them on opposite sides of the ladders, and then your ladder